For our genetics labs, we are going to be performing something called a chi-squared test. The notation for the name of this test is a cursive x squared. This does not mean x squared. The x squared here stands for the name chi-squared. So what we're going to do with this test is use it to determine if the data that we see has varied from the genetic ratio that we expect from our parental process. The chi-square test is called the goodness of fit test, and basically what it's looking for is whether or not your observed ratios are a good fit for your expected ratios. So the actual ratio of the phenotype that you see may not be exactly what you expect from performing a Punnett square. Part of the reason is that a Punnett square gives us exactly 4 outcomes, or exactly 16 outcomes of different genotype combinations, and therefore a limited number of phenotype combinations. In any parental pairing, we may not get exactly 4 offspring or exactly 16 offspring. We might get a number that's not even a multiple of 4 or 16. We might get 247 corn kernel seedlings, or we might get 7 children or 3 children. And so even though we are expecting, say, a 3-to-1 ratio in our offspring, we may not see it simply because we don't have the exact number of outcomes possible. The other possibility is that every single combination of egg and sperm is a chance event. So it's basically a flip of a coin which sperm is fertilizing which egg. And so if we think about genetics as a flip of a coin, and we think about flipping a coin that is heads and a coin that is, a coin with heads and a coin with tails on the other side, we would expect a 50-50 heads and tails outcome. So if we flip that coin 100 times, we can reasonably expect we're going to get 50-50. But if you do this, you're probably not going to get exactly 50-50. You might get something like 49 and 51 or 45 and 55. And you might think, that's weird, but that was just chance. The exact same thing happens with genetics. What we often see is that our ratios don't exactly fit our expected observed outcome simply due to chance. And so the goodness of fit test is going to tell us when our observed data ratios are a good fit for our expected ratio. So what this means is it can tell us that, hey, 48 and 52 outcomes for flipping a coin is not weird. But if, say, we got 90 heads and 10 tails, we might start to think there's something wrong with the situation because that outcome, although it can happen, is very unlikely. And so the chi-square test is going to help us determine when we fall into that range of, you know what, that has deviated way too much from what we expect. Therefore, we're going to say that we have not followed our ratios. So if our deviation from what we expect is relatively small, you can probably assume that it's just a chance event. We flipped it 100 times and we didn't get exactly 50-50. But if your expected ratio is really large, then you might start to think that something is going wrong. If we think of this coin flipping, maybe our coin is not weighted correctly and something's causing the heads to constantly land on top. Or maybe there is something else that is affecting the outcome. So the chi-square test is a calculation that we can do, an actual value with a cutoff point that we can say, if we get this value, then we know that our deviation from what we expected is really big, and it's pretty unlikely that that just happened, and so there must be something interfering. So we're going to walk through how we actually use this chi-square test now that we understand what it does. Our chi-squared formula is at the top. Again, we've got that x. That does not mean x squared. That's telling us our chi-squared value. And we need some information in order to calculate this. First, this symbol here, that indicates that we're going to take the sum. So we are going to take the sum of a set of these d squared over e values. In your lab, you have a fancy little chart that's going to help you do this really, really easily. E stands for our expected value. This is going to come from the Punnett square and depends on our number of offspring and expected outcome. So this is a value that we're going to have to figure out, because remember, our Punnett square is going to give us a value out of 4 options, but we're very likely going to have more than 4 options. O is the observed value. This is the actual value that you observe. So it is very likely not going to be out of 4 total options. D is a value called deviation. This is our deviation from what we expect to happen. We calculate this by taking our observed value O and subtracting our E value, our expected value. So D is an additional step we're going to have to calculate. Again, we've got a nice little table for you. In order to figure out if our chi-squared value is significant, we need to know the number of classes, which is the number of possible outcomes. From that, we also need to know something called the degrees of freedom. Degrees of freedom are equal to your number of classes minus 1. This sum value says that we're going to calculate all of our d squared over e values and then add them all together. We're going to go through an example from our Biology 101 lab. In this lab, we cross some corn plants that have genes for production of chlorophyll. Now, production of chlorophyll is the dominant gene. We're going to denote that by big G. And an albino plant who doesn't produce chlorophyll is the recessive. So we're going to cross 2 parents who are heterozygous, and we're going to look at the outcome. The actual plants that we see is the outcome of our cross, and they are going to be our expected values. So let's say that we observe 33 green seedlings and 7 albino seedlings. So this is a total of 40 seedlings altogether. In order to determine our chi-squared value, we need to figure out how many we would expect to be green and albino if we had 40 total seedlings. So this is our observed value. And we need to figure out how many we would expect. So to do this, to get our expected value, we're going to perform a Punnett square with these parents. So I'm just going to go ahead and fill out the Punnett square. And we are going to get 3 green to 1 white. In other words, 75% of our total of 40 should be green, and 25% of our total of 40 should be white. So to get our expected values, we are going to multiply our 40 total seedlings by 3/4 to get our number of green. So that should be 30. And then we're going to calculate our white by multiplying 1/4, because 1/4 of them are going to be white, times our total number of 40, which means we are going to have 10 white. So if we put that into our chart, we expect 30 green, 10 white. We actually observe 33 green and 7 white. Our deviation, our D value, is equal to observed minus expected. So we're just going to take our observed value 33 minus 30 to get a value of 3. For our white individuals, we take our observed 7 minus our expected 10, which is -3. Just leave the negative there, doesn't matter. It's just telling you that you have 3 more and 3 less than you expected. Then we're going to calculate D squared. We're just going to square 3 and -3 to get 9. Then we're going to calculate D squared minus E. So for the first column, we're going to take our 9, our D squared, divided by E value of 30. And it's going to come out to 0.3. For our second column, we're going to take our d squared 9 divided by 10 to get 0.9. Then the sum part, we are going to sum. We're going to sum up our d squared over e values. So we're going to take 0.3 plus 0.9 to get our chi-squared value. So our chi-squared value equals that sum, which is 1.20. We're not done yet, but that chi-squared value is super important. The other thing that we need is our degrees of freedom. So degrees of freedom equals the number of classes minus 1. So we have green and albino, that's 2 classes minus 1. So our degrees of freedom equals 1. We need that for our chi-squared determination of the probability of this outcome actually happening. Our chi-squared value is going to be affected by several different things. It's affected by how much deviation you actually have. It's affected by the sample size, so we're going to have the potential for much more deviation in a small sample. And then it's also affected by how many different outcome options there are or number of classes. So the chi-square test answers 2 questions. One is how probable is it that the deviation that you have occurred, and are the results close enough to what you expect? When we perform a chi-squared test, we are trying to defend a hypothesis. This is not your experimental hypothesis. This is a statistical hypothesis. The statistical hypothesis is called the null hypothesis, and it basically says that there's no deviation from what you expected, and that any deviation that you had is just due to chance, kind of like flipping that coin 48 times on heads and 52 times on tails. Just by chance, you deviated a little bit. So we make a specific statement about this. When you write your hypothesis, it should be specific to the observed and expected values that you expect to see. So in the null hypothesis, we are going to state that any deviation from an expected value is due to chance, and that there is no statistical difference between the observed and expected ratios. So when you write your null hypothesis, you want to add in there what your expected outcome is. So for our test, our expected outcome is the 3-to-1 ratio. So the basis for your null hypothesis is going to be the same every time, except that you're going to add in what you expect to see. So we have determined our expected values, we've counted up our observed values, and we've calculated our chi-squared values. We needed our degrees of freedom, so our degrees of freedom is 1, and our chi-squared value is 1.2. So the first thing we need to do is use our degrees of freedom to tell us which line in our chi-square table we're going to use. It tells us that in this first box there. So degrees of freedom is the first column, or degrees of freedom equals 1. So that means that we are going to use this first row. So our chi-squared value is going to fall somewhere in this row. So we're going to look for that chi-squared value of 1.2. In our first row, our chi-squared value is closest to 1.074, but it hasn't quite reached 1.642. So what this tells us is that the probability, that's our p-value, the probability that we get this outcome, this observed outcome, is between 30 and 20%. So the p-value is going to tell us the probability of our outcome. That's a little weird to think about it because we're thinking about the probability that we're going to get these results instead of the probability that the results demonstrate. It's a little bit weird. but this is the probability that if we performed this exact test 100 times, we would get this result. So what this tells us is that roughly 30% of the times we do this test, we're going to get this result. If we have a p-value that occurs on the left-hand side, like ours, it tells us that our deviation is insignificant. In other words, we do not need to doubt that our null hypothesis is correct. So we have no reason to reject our null hypothesis. In other words, we are saying with this p-value that there's a good chance we've got this data, and so we're not going to worry about it. That data is close enough to our expected value. If you find a p-value on the right-hand side, it tells us that our deviation is significant. And that tells us that our observed ratios are not close enough to our expected. And therefore, we have to reject the null hypothesis. And say that our observed values are not what we expected. So let's do another example. This is just giving you the chi-squared value, not the calculations, for a 4 class. So this would be for a dihybrid cross where we have 4 possible outcomes. And let's say that our number of classes is 4, which makes our degrees of freedom 3. So we're going to use row 3. We're going to look for this 8.123 value in our third row. So if we look in our third row, that 8.132 falls on the right-hand side. It falls between a p-value of 0.05 and 0.02. So we're going to say our p-value is between, let me write it correctly. Our p-value falls between 0.05 and 0.02. When our p-value falls on the right side, our deviation is significant. This means that we have reason to doubt our null hypothesis, that we have deviated too much from what we expect, and we need to reject that null that states that our observed is the same as our expected. Again, if it would have fallen on this side, then we have no reason to doubt that null hypothesis, and we do not reject that null hypothesis. This slide is just a summary of the conclusions that you make based on your p-value. Go ahead and write this down in your notes so that you have it for analyzing your data later.